Welcome back. Today, we're jumping right into this explainer, cracking open some really fascinating alternative medical literature. We're going to map things out, kind of like flipping through a scientific sketchbook, to explore a totally different way of looking at cellular health and disease. Specifically, we're talking about alternative metabolic theories, cellular pH, and these things called mineral transporters, like lithium orotate.
So think about this for a second. What if, instead of just focusing heavily on genetics, we ask, what if cancer is actually fundamentally a cellular environment problem? We're going to unpack the ideas of researchers who hypothesize that the absolute root of the disease lies in the basic electrical and chemical balance of the cell itself. It's all about changing the terrain of the body, making it literally inhospitable to disease.
Okay, the crucial starting point here is something early researchers, like Dr. Max Gerson, noticed. They found that cancer is often tied to a major imbalance in a cell's sodium-potassium gradient. Now, you can think of this gradient as essentially the battery that powers a cell's daily functions. It maintains the vital electrical charge across the membrane. When that gets disrupted, the cell's natural charge is totally compromised. The literature basically suggests that if you could find a medicine to stimulate and fix this gradient, recharge that battery, so to speak, it would essentially be Gerson therapy in a pill.
To really picture this, imagine drawing a line right down the middle of a board. On one side, you've got a thriving, healthy cell. It's loaded with potassium, it's highly alkaline, and it's rich in oxygen. But on the other side, the cancer cell is a drastically different story. These cells survive by using a fermentation process to produce energy, a totally anaerobic oxygen-free pathway. And you know what that fermentation creates? Lactic acid. That leads to a highly acidic environment. Plus, cancer patients almost always show a massive excess of sodium in their blood. So it's this incredibly toxic, high sodium, highly acidic, low oxygen state.
Right, so how do we fix that? To correct this cellular imbalance, to push out all that excess sodium and drag the right nutrients back in, researchers realized they needed a delivery vehicle. And that brings us to mineral transporters. You can think of these as tiny, highly specialized cargo ships. They're designed to actively carry specific minerals straight through the cell's tough outer membrane. So instead of just floating around uselessly in the bloodstream, these transporters actively penetrate the cell and drop their payloads exactly where they're needed, like right into the mitochondria, the actual powerhouses of the cell.
A major pioneer in this whole alternative framework was Dr. Hans Nieper. Back between 1958 and 1960, while he was a research fellow at the Paul Ehrlich Institute in Frankfurt, he actually started developing these specialized mineral transporters. He was essentially engineering microscopic chemical keys to unlock the cell membrane. His entire goal was to find a safe, effective way to transport those vital, charge-restoring minerals straight into the heart of the cell, to fix the exact environmental imbalances we were just talking about.
And here is where it gets really interesting, because Dr. Nieper actually drew a lot of his inspiration straight from nature, specifically deep-sea sharks. He took note of a massive investigation by the Smithsonian Institution where, get this, out of roughly 25,000 sharks studied, they found exactly one single malignant tumor. One. Sharks almost never get cancer, which is a stark contrast to other fish that frequently develop tumors when swimming in polluted waters. This wild statistic pointed Nieper's attention directly at the shark's unique biology for cancer resistance.
That incredible observation formed the bedrock of his theory. See, he figured out that sharks can actually recycle ocean salt back into the sea thanks to specific agents produced in their livers, effectively dumping excess sodium out of their cells. He theorized that squalene and other compounds in shark liver oil could trigger that exact same sodium elimination process in human cancer cells. To replicate and even supercharge this targeted sodium dumping effect, he prescribed specific mineral transporters, most notably a compound called lithium orotate, literally to force that superfluous sodium right out of a patient's cells.
When putting this into practice, Dr. Nieper reported in his clinical notes using daily dosages of 150 to 450 milligrams of lithium orotate. And the crazy thing? He claimed the toxic side effects at those doses were practically non-existent. Because lithium orotate acts as such an insanely efficient mineral transporter, it gets absorbed directly into the cells. According to Nieper's protocols, this direct, deep cellular absorption meant you didn't even need routine blood tests to monitor lithium levels in the bloodstream, because it wasn't pulling there. It was going right to work inside the cell.
Now let's zoom out a bit and see how this fits into the broader strategy of cellular alkalization. Using transporters like lithium orotate is really just one crucial piece of a much larger puzzle.
The ultimate goal here is to completely reverse that acidic environment that lets cancer cells metabolize and multiply in the first place. By forcefully pushing out sodium and pulling in the right elements, the strategy is to systematically dismantle that acidic, fermenting terrain, replacing it with a highly alkaline, oxygen-rich environment where healthy cells can thrive and cancer cells literally cannot survive.
According to these alternative frameworks, there's a specific crew of alkalizing minerals that act as the chemical tools to neutralize this acid and restore balance. We're talking about minerals like calcium, magnesium, potassium, and zinc, which are considered highly cancer protective. Magnesium, for instance, is specially noted for neutralizing the acid produced by the cancer process itself. Interestingly enough, isotope studies have shown that potassium, rubidium, and especially cesium are incredibly efficiently taken up by cancer cells. But the literature makes it very clear, these minerals have to work together as a team. Missing even one can cause the whole cellular operation to malfunction. So why go through all the trouble of flooding the cells with these specific highly alkaline minerals? Well, by using these precise combinations and transporters, the ultimate endgame of these alternative therapies is to drive the cell's internal pH up to a very specific target, the level of 8. In this model of cellular health, hitting that number 8 is the absolute critical threshold. Once you reach that exact pH, the entire metabolic behavior of the cell is forcibly changed. There's a brilliant quote from Dr. A. Keith Brewer that encapsulates this theory perfectly. Based on his mass spectrographic studies, he stated that the quantity of cesium taken up was sufficient to raise the cell to the 8 pH range where cancer cell mitosis ceases and the life of the cell is short. Essentially, flooding the cell with these alkaline minerals forces a state that completely halts mitosis, which is the division and growth of the cancer cells. Once that internal pH hits 8, the lifespan of the cancer cell drops dramatically. It's quite literally a targeted environmental attack from the inside out.
Believe it or not, this entire approach traces its roots all the way back to a foundational discovery by Nobel laureate Dr. Otto Warburg. He was the one who originally discovered the absolute baseline of this cellular dysfunction. Warburg found that lowering tissue oxygen levels by just 35% for a mere 48 hours is enough to convert normal, healthy cells into irreversible cancer cells. The cells are basically forced to adapt to survive, switching over to that exact acidic fermentation process we mapped out earlier. So everything we've looked at today, from dumping the sodium to using mineral transporters to driving up alkalization, it's all one big coordinated attempt to completely reverse that exact Warburg effect.
We've traced the lines from cellular voltage and the sodium-potassium battery all the way to specific mineral transporters like lithium orotate, and finally to the grand strategy of cellular alkalinization. And it really leaves us with a massive, compelling question. Could the true future of oncology lie in fundamentally rewriting the environment of the cell itself? If cancer is just as much a metabolic and environmental issue as it is a genetic one, then tools that actively alter the cellular terrain might just be the most important notes we have in the scientific playbook. So what do you think? Thanks for joining me on this explainer, and definitely keep asking those big questions.